Let's talk about cancer diagnostics. Every year, thousands of patients get their tumors sequenced using FDA-approved NGA panels. The idea is simple: detect mutation, match it to a drug, and start the treatment. But the reality, the clinical benefit is just 5 to 15%. Let's unpack why. Current FDA-approved diagnostics rely on static mutation detection. Whole genome sequencing, whole exome sequencing, or gene panels pick up genomic variants like EGFR, BRAF, or ALK fusions. Companion diagnostics, the FDA-approved tests, work like a lookup table. If the mutation is present, you are matched to the drug. For example, EGFR mutation means osimertinib. The underlying assumption is straightforward. The mutation itself drives the cancer, and blocking it with the right drug should help the patient. But that assumption holds good only in some cases. Now let's look at how these drugs are developed. Preclinical work doesn't happen on real patient tumors. Instead, scientists engineer models like CRISPR-edited immortalized cell lines, organoids, even animal models. These systems are used to validate if we block this pathway, does the tumor shrink? If yes, the drug moves forward into clinical trials. Eventually, FDA approval is granted based on success across groups of patients, not on case-by-case functional readout. So the entire drug discovery pipeline is built on models that only approximate patient biology. But here is the problem. Real biology is far more complex. Take the exact same mutation, say EGFR. In one patient, a smoker, the mutation might sit alongside multiple other co-mutations and inflame tumor microenvironment. In another patient, a non-smoker, the biology looks completely different. The same mutation leads to different consequences. On top of that, we have transcriptome shifts, epigenetic changes, stromal interactions, and immune environments, all of which shape how a tumor responds. This explains why, even for so-called perfectly matched drugs, clinical benefit rates remain only in the 5 to 15% range. And that brings us back to the numbers. Large trials like Shiva, Moscato 01, iPredict, and NCI Match have shown the same pattern. The actual benefit rate from NGS-guided therapy is only 5 to 15%. Why so low? Because mutations do not equal actionable biology. A genomic match doesn't guarantee functional dependence. Tumor heterogeneity creates multiple competing clones. NGS is static. It doesn't tell us pathway activity in real time. Targeted drugs are often only effective in narrow FDA levels, and tumors evolve, leading to resistance. So what looks like a neat lookup table, mutation to drug, breaks down in the complexity of real patient biology. So yes, the picture is clear. FDA-approved NGS diagnostics are essentially static mutation to drug lookup systems. They are based on engineered models, validated across populations, but not tuned to the unique biology of each patient. And while the science behind them is powerful, the clinical reality is that the majority of patients don't benefit, because the real story of cancer isn't written in static code. It's written in how cells behave. And that's where the future begins. In the coming posts and videos, we will explore what it looks like when we move beyond static DNA and start capturing live patient biology in action. Stay tuned.